Advancing CAR-NK Therapy, Why Leukopak Quality and Donor Characterization Matter. The success of chimeric antigen receptor CAR T-cell therapy has driven interest in applying CAR technology to other immune cells. Natural killer cells are emerging as a promising platform for adoptive cell therapy. NK cells can be equipped with CAR machinery for cancer retargeting and enhanced cytotoxicity in much the same way as with T cells. As part of the innate immune system, NK cells can recognize and eliminate malignant cells without prior antigen sensitization or major histocompatibility complex engagement. They mediate cell death through multiple mechanisms, including direct cytotoxicity via the release of cytolytic proteins, antibody-dependent cellular cytotoxicity, and can activate the adaptive immune system to amplify the immune response. Many CAR-NK programs rely on leukopak collections as a key starting material for isolating NK cells. There are several advantages to CAR-NKs over CAR-Ts that have the field excited. Broad anti-tumor activity. In addition to cytotoxicity driven by the engineered CAR, NK cells retain their native receptor functions. Lower toxicity. Short-lived persistence reduces the risk of toxicities commonly associated with CAR-T therapy, such as cytokine release syndrome and graft versus host disease. Off-the-shelf potential. CAR-NK activation is MHC independent, overcoming HLA matching requirements that limit some allogeneic CAR-T approaches. As with CAR-T development, leukapheresis collections are relied upon for CAR-NK development too. The quality and consistency of these starting materials are important, as variability impacts quality attributes that affect manufacturability and clinical outcomes. This variability can come from the collection and processing methods and from the healthy donors themselves. Vendors with robust donor characterization protocols, including demographic screening and cellular attributes such as HLA typing and KIR profiling, provide researchers with critical data to optimize donor selection and reduce variability. And for clinical applications, regulatory compliance must be considered. Donors for GMP collections must meet stringent FDA criteria, and have a vendor with a track record of success in qualifying these donors is critical to translational success. Because of the allogeneic potential of CAR-NK cell therapy, ensuring the quality and consistency of starting material supply from preclinical to clinical is even more critical. Working with an experienced vendor who can provide both RUO and GMP leukopaks can mitigate delays in clinical translation of CAR-NK candidates. This can improve timelines and cost delays that could arise from having to revalidate source material and suppliers. This also provides the flexibility and agility to bilaterally transition between preclinical and clinical phases to address the dynamic nature of cell therapy product development. Other factors such as accredited facilities, trained staff, optimized processes, and a robust quality and GMP framework are also important differentiators across vendors. Beyond cell therapy, high-quality leukopaks also serve as the starting material for ex vivo gene editing applications, where quality and consistency are just as essential. In our next blog, we'll explore how leukopaks are driving innovation in allogeneic gene editing strategies and enabling the next generation of cell and gene therapies.